Okay, let's unpack this. Welcome to the deep dive, where we really try to get into the weeds on the research, pull apart these complex mechanisms, and hopefully break it down into insights you can actually use.
That's the goal.
Today, we're tackling something I find genuinely fascinating, a real biological paradox. We are diving deep into Kisspeptin-10. It's a peptide, right? And it's tiny. Just 10 amino acids long.
Incredibly small.
Yet somehow, it acts as this master regulator for some of the absolute biggest events in, well, human life. We're talking puberty, fertility, even mood and metabolism.
It really is a remarkable molecule. Its influence is just vast, completely belying its size. And our mission today really is to unpack the source material we've looked at.
Right.
We want to reveal how this single powerful signal dictates precisely when and how the human body decides, okay, now I'm ready to reproduce.
Yeah.
And how it connects that physical readiness to our emotional state, and also how deeply it's integrated with our overall energy status. It's like a metabolic checkpoint.
A checkpoint. Yeah, I like that.
Yeah.
And here's a fascinating little nugget to start us off. Something from the history that really shows the strange path science takes sometimes.
Whoa.
Kisspeptin. It comes from a larger protein coded by the Kisdalis-1 gene.
Mm-hmm.
Now, you'd think, given what we know now, that it was named for, reproduction.
Yeah.
Maybe kissing.
You would think so, yeah.
But no. When it was first found back in the 90s, the researchers weren't looking at fertility at all. They were studying cancer.
It was initially found in melanoma cells, wasn't it?
Exactly. So in this kind of beautiful, almost accidental twist, it was originally identified and named as a tumor suppressor.
A metastasis suppressor, specifically.
Its profound, absolutely fundamental role as the reproductive master switch. That was a completely unexpected finding decades later that just revolutionized neuroendocrinology.
And that historical context is really important, I think. It shows us this molecule's reach goes way beyond just the hypothalamus. Its original job, or discovered job, inhibiting cell growth in cancer cells, it points to a general power over cellular processes, a potency that the brain then sort of co-opted, you could say, to regulate the life cycle itself.
Okay, so let's zero in on the basics then. What exactly is Kisspeptin-10? We know it's small, but what's its structure? Where does it come from precisely?
Right. So structurally, Kisspeptin-10 is what we call the bioactive core of the larger Kisspeptin protein. It's a short chain, literally just 10 amino acids linked together in a specific sequence.
And it's snipped off from the bigger molecule.
Exactly. It's cleaved from the much larger parent molecule. But the key insight here, and it's why we often focus specifically on the naganeptin fragment, is its incredible potency. This tiny piece retains full biological activity.
Wow.
It's the absolute minimum structure needed to do the job. Anything less, and it doesn't work.
So it's super efficient. And where does it actually do its main job? If it's controlling the whole reproductive system, where's that central control panel?
That panel is located deep in the brain, specifically in the hypothalamus. And it works by binding to a very, very specific receptor protein called Kiss1R. Think of it like a lock.
A Kiss1R lock.
Exactly. And this receptor, this lock, is found almost exclusively on these critical neurons in the hypothalamus that are responsible for regulating reproduction. It's highly targeted.
I think an analogy might help here because this is just so fundamental. Let's imagine the reproductive system is like a huge, complex power grid.
Okay.
Kisspeptin-10 isn't the whole grid, obviously. It's more like the key, the only key.
Mm-hmm.
It's the specific key that fits perfectly into that Kiss1R lock, which is like the main power switch in the central control room.
Right.
If you don't have that key or you lose it, the entire system just stays dark. It stays off. Doesn't matter what else is going on.
That's a perfect analogy. It holds up really well.
Yeah.
Because the moment Kisspeptin-10 engages that lock, that Kiss1R receptor,
Yep.
It immediately initiates this fundamental multi-step chain reaction. It's what we call the hormonal cascade, and it's really the heart of timing human reproduction.
Okay, let's trace that cascade step by step. What happens right after Kisspeptin-10 binds to Kiss1R? What's the very first domino?
Step 1, that binding event triggers the activation of those specialized neurons we mentioned specifically, the GnRH neurons.
GnRH, gonadotropin-releasing hormone.
Correct. The activation forces these neurons to release GnRH into this special little network of blood vessels connecting the hypothalamus directly to the pituitary gland just below it.
And GnRH is often called the master hormone in this context, right?
It is, yeah. You could call it the big boss hormone because it's the critical initiating signal for the entire reproductive axis, the HPG axis, hypothalamic pituitary gonadal.
Now, something I find fascinating about GnRH, and this really highlights kisspeptin's precise control, is that it's not just dumped out continuously. It has to be released in pulses.
Absolutely critical. It must be released in these rhythmic pulses. The frequency of the pulses, how often they happen, and the magnitude or amplitude, how strong each pulse is, that pattern is absolutely vital.
For what? For development, fertility.
Both. For normal development through puberty and for sustained fertility in adulthood. If that pulse pattern is wrong, too fast, too slow, too weak, too strong, or just erratic, the whole system downstream starts to fail.
Okay, so GnRH is released in pulses, travels down to the pituitary.
Correct. Through those portal blood vessels, it gets there very quickly.
And that triggers step 2.
Step 2. The pituitary gland receives these rhythmic commands from the hypothalamus via GnRH, and in response, it releases the 2 primary gonadotropins into the general circulation.
LH and FSH.
That's them. LH, luteinizing hormone, and FSH, follicle-stimulating hormone. These are the real workhorses that travel throughout the body to the main targets, the gonads. So ovaries in women, testes in men.
Right, and that gets us to step 3, where LH and FSH have their specific jobs. They don't just do the same thing, do they?
No, they have distinct, though coordinated roles. In both men and women, LH is the primary driver for producing the sex steroids.
Testosterone in men.
And estrogens and progesterone in women. LH also delivers the critical signal, that big surge, which triggers ovulation in women.
Okay, so LH is about the hormones and the final trigger. What about FSH?
FSH is more the maturation hormone. Think follicle stimulating. In women, it's crucial for developing the ovarian follicles, which contain the eggs, getting them ready.
Makes sense.
And in men, FSH is vital for supporting sperm development, the process called spermatogenesis.
So, working together, LH handles the hormonal environment and the big events like ovulation, while FSH focuses on making sure the eggs and sperm are developing properly.
You've got it. And this entire incredibly complex, finely balanced system, it all hinges on that tiny 10 amino acid peptide, Kisspeptin-10, initiating that very first rhythmic release of GnRH from the brain. It's quite something.
Okay, now we understand the mechanism, the cascade. Let's look at its impact across a person's life. The sources we looked at strongly suggest kisspeptin isn't just involved in puberty. It seems to be the essential signal that actually allows it to happen.
That really is the current understanding, yes. It's thought that the gradual ramping up of kisspeptin signaling within the hypothalamus, something that happens slowly, steadily over years during childhood, is the definitive key that unlocks puberty.
So the system is kind of dormant in childhood.
Exactly. It's in a state of quiescence or silence. For the reproductive system to wake up and transition to adult activity, those GnRH pulses we talked about have to reach a critical threshold, both in frequency and strength. And kisspeptin appears to be the primary upstream signal that drives that functional shift, that increase in GnRH pulsatility. Without kisspeptin signaling increasing and then maintaining its pace, puberty just doesn't happen.
That level of control has some pretty immediate and profound clinical implications, doesn't it? If someone has delayed puberty or it never starts, this points right to a potential failure point. What does that look like clinically?
It typically manifests as a condition called hypogonadotropic hypogonadism, often shortened to HH. And we see these rare but incredibly informative genetic conditions where individuals have severe mutations, either in the kisspeptin gene itself, KISS1, or in the gene for its receptor, KISS1R.
So the key or the lock is broken.
Essentially, yes. In these individuals, the entire reproductive axis remains silent. The brain never sends the proper signals to the pituitary, so the pituitary never signals the gonads. The body is effectively stuck in a prepubescent state.
Which leads to infertility and lack of secondary sexual characteristics, I assume?
Correct. It prevents the development of those characteristics and results in infertility.
That's obviously devastating for the individual.
Yeah.
But from a scientific standpoint, it's a powerful validation, isn't it? If the problem is the switch is broken, then maybe providing the key could fix it.
And that's exactly where some really incredible therapeutic developments have occurred. Using Kisspeptin itself or synthetic analogs that activate the KISS1R receptor.
You can give it as a treatment.
Yes, in many patients with HH caused by defects in this pathway, providing exogenous Kisspeptin can actually flip that light switch on. It allows their own hypothalamic pituitary gonadal axis to finally respond, initiating puberty or restoring reproductive function. It really confirms Kisspeptin as that non-negotiable fundamental trigger.
Nature loves balance, though. We have to consider the flip side. What if there's too much Kisspeptin signal, or it starts too early?
Good point. And the sources do indicate that. Excessive or premature Kisspeptin activity, maybe from certain genetic variations or other forms of overactivation, has been linked to the opposite problem, precocious puberty.
So puberty starting way too early.
Exactly, which just reinforces its role. It's not just an on-off switch, it's also the main volume control and the timer for the whole process of sexual development.
Okay, so it gets puberty started. What about after that? How crucial is Kisspeptin-10 for regulating fertility throughout adult life? Does its job kind of taper off once the system is up and running?
Not at all. It doesn't taper off. Its role just shifts slightly. It transitions from being primarily the starter signal to being more like the conductor of the orchestra. It remains absolutely essential for regulating the continuous rhythm, that pulse pattern of GnRH release throughout adulthood. Remember, that precise pattern is critical. A continuous, non-pulsatile flow of GnRH would actually shut the system down through receptor desensitization.
Right, the pituitary would just stop listening.
Exactly. So Kisspeptin ensures that necessary pulsatile pattern is maintained day in, day out, month after month.
How does that pulsatility, that rhythm section, play out differently in women and men?
Well, in women, the role is highly dynamic, very cyclical. While it maintains a sort of baseline pulse rate, there's a major, dramatic, and very carefully timed surge in Kisspeptin levels just before ovulation.
Ah, okay.
This Kisspeptin surge is thought to be the key event that precedes and helps trigger the big preovulatory surge of LH from the pituitary.
Which is the direct trigger for the egg being released.
Precisely. So Kisspeptin is a critical step in supporting that final maturation of the ovarian follicles and ensuring the timely release of the egg each month.
And what about in men? Their hormonal profile is generally more stable, less cyclical.
For men, it's more about continuous support. Kisspeptin signaling maintains the necessary GnRH pulsatility required for steady, robust testosterone production by the testes and for ongoing, continuous sperm development.
So keeping the factory running smoothly.
Essentially, yes. And, as we'll get into a bit more later, there's also evidence suggesting Kisspeptin signaling continuously influences libido and sexual behavior in men too, providing that constant link between physical ability and the drive to act.
Okay, this is where things get really interesting in terms of application. Innovation in fertility treatments. Our sources mentioned using Kisspeptin-based approaches in IVF. What's the big advantage there compared to what's normally done?
Right, this is a really promising area. So the current challenge, one of the key steps in IVF, is triggering the final maturation and release of the collected eggs.
Okay.
Clinicians often use injections of a synthetic hormone called HCG, human chorionic gonadotropin. It mimics LH effectively.
There's always a but.
But HCG is a very powerful, somewhat blunt instrument. While it works, it carries a significant risk of a nasty side effect called ovarian hyperstimulation syndrome, or OHSS.
OHSS, that sounds not good. What is it?
It's basically the body's overreaction to the strong stimulation. Because HCG hits hard, it can cause the ovaries to produce way too many follicles, leading to significant ovarian swelling, fluid shifting out of blood vessels into the abdomen. It can be quite serious, even dangerous, potentially causing things like blood clots.
Yikes. Okay, so how does using Kisspeptin help avoid that?
Kisspeptin offers a much gentler and, crucially, more physiological trigger. Instead of bypassing the brain and hitting the ovaries directly with a powerful synthetic hormone,
You use the body's own system.
exactly, you use Kisspeptin-10, or an analog, to activate the patient's own hypothalamus-pituitary axis. You're using the body's natural master switch to signal the release of its own LH, typically in a more controlled natural surge pattern.
Ah, so it's less like a sledgehammer and more like
Right.
nudging the natural system to do its thing.
Precisely. This approach significantly reduces the risk of OHSS because you're less likely to get that overwhelming, uncontrolled ovarian response. It allows for a potentially safer, perhaps more predictable, final egg maturation process in IVF.
That seems like a really significant step forward for patient safety in IVF. Using the body's own exquisite control system rather than overriding it. Okay, if Kisspeptin-10 was only about puberty and fertility, that would be huge enough. But the research, the molecular geography, tells us there's clearly more going on. Let's talk about this integration with emotion and mood. That's quite profound.
It really is. This is probably one of the most exciting frontiers in neuroendocrine research right now, because Kisspeptin and its receptor, Kis1R, they aren't just stuck in that core reproductive control center in the hypothalamus.
Where else are they found?
They're also found in significant amounts in other brain regions, heavily involved in processing emotion, regulating mood, social bonding, and even sexual behavior. Areas like the amygdala, parts of the limbic system.
The emotional brain.
Exactly. So its physical location in the brain strongly suggests Kisspeptin acts as a key integrator. It seems to bridge our physical readiness to reproduce with the necessary emotional and behavioral states for things like mating, attraction, and bonding.
So what happens when researchers actually stimulate Kisspeptin signaling? What effects do they observe?
Well, studies, both in animals and increasingly in humans, have shown some really tangible psychological and behavioral effects. Activating Kisspeptin pathways can demonstrably enhance positive mood, increase alertness. It can also increase self-reported sexual desire or libido. And perhaps most intriguingly, it seems to modulate how we respond to romantic or emotionally charged stimuli. It might make individuals more sensitive or responsive to those kinds of interpersonal cues.
Wow, so it's sitting right at that intersection between raw biological drive and our capacity for emotional response and connection.
That seems to be the case. It's fundamentally linking the two.
That's the really profound implication here, isn't it? Biology isn't just flipping a switch for, say, testosterone release or ovulation. It's also actively shaping the feeling, the motivation, the behavioral drive that's needed for intimacy, for pair bonding, for connection. You can't really reproduce effectively without the motivation and the whole emotional context.
Absolutely. It strongly suggests that Kisspeptin isn't just a simple hormone release factor. It's a molecule that actively promotes the entire suite of conditions, physical, emotional, behavioral, associated with successful reproduction. Biology seems to demand that the body is not just capable of reproducing, but also motivated and behaviorally ready to do so. Kisspeptin seems central to making sure all those pieces are aligned.
Okay, let's pivot slightly to the body's energy status. You mentioned earlier, Kisspeptin is really sensitive to nutrition and stress. How does it act as this energy balance sentinel? What signals is it actually listening to?
Right. This is one of its most vital and probably most ancient protective roles. Those Kisspeptin-producing neurons, particularly in the hypothalamus, are exquisitely equipped to monitor the body's overall metabolic health.
How do they do that?
They have receptors on their surface for key metabolic hormones, hormones like leptin, for instance.
Leptin comes from fat cells, signals fullness, right?
Exactly. Leptin signals long-term energy stores, basically telling the brain how much body fat is available. They also have receptors for ghrelin.
The hunger hormone?
Right. Ghrelin signals hunger, acute low energy. And they're also sensitive to stress hormones like cortisol.
So these neurons are getting constant metabolic reports, like updates from the body's energy accountant.
That's a great way to put it. Constant updates. And if the body senses a significant energy deficit, maybe very low body fat, so low leptin, or if it's undergoing intense physical stress like extreme endurance exercise or acute illness or severe dieting, which might mean high ghrelin and high stress hormones.
Basically, if the energy reserves are flashing red.
Precisely. If the overall message from these metabolic signals is warning, insufficient resources available, then Kisspeptin signaling drops.
Yeah.
Sharply.
Okay. And when that metabolic red flag goes up and Kisspeptin levels plummet, the protective shutdown mechanism kicks in.
That's exactly what happens. The GnRH pulses slow down dramatically or might even stop altogether. And consequently, the entire reproductive system downstream effectively shuts down.
Right.
It's the body making a very clear survival-oriented calculation. All available energy is needed for basic maintenance and survival right now. This is absolutely not a good time to undertake the huge energy demands of pregnancy or sustained reproduction.
This provides such a clear molecular explanation for that well-known phenomenon, functional hypothalamic amenorrhea.
Yeah.
Especially common in female athletes, right?
Absolutely.
Like endurance runners, gymnasts, dancers.
Yeah.
Often very low body fat, very high training stress, and they stop menstruating.
That is the classic real-world example of this system in action. It's the brain sensing that chronic low energy status, interpreting it via the suppression of Kisspeptin pathways, and dialing down the reproductive drive as a protective measure. It's a primal survival mechanism.
And presumably, if energy balance is restored.
Yes. Once energy stores are replenished, body fat levels normalize, maybe training stress reduces, the suppressed Kisspeptin activity typically resumes, the GnRH pulses restart, and the reproductive system switches back on.
Fascinating. Okay, now we do need to touch on the regulatory side of things, especially concerning sports and the wider research chemical world. Our source material was very clear. Kisspeptin-10 is on the anti-doping list. Why? If it just stimulates your own natural hormones.
It's because of its sheer biological efficiency and power.
Yeah.
When someone administers exogenous Kisspeptin-10, it causes a massive and very rapid surge in LH levels from the pituitary, much higher than normal physiological peaks. And that huge LH surge, in turn, drives a sharp, often supraphysiological meaning above the natural range spike in the body's own production of testosterone.
So the potential for misuse is obvious. An athlete might be tempted to use it as a kind of backdoor testosterone booster,
Exactly.
stimulating their own production to get an edge, maybe thinking it's harder to detect than taking synthetic testosterone directly.
That's precisely the concern. Its natural mechanism appeal could make it seem attractive for misuse. However, artificially generating those extremely high levels of LH and the resulting testosterone surge is absolutely considered performance-enhancing and creates an unfair advantage. That's why it's now officially classified as a prohibited substance by WADA and other major anti-doping agencies.
Right, and this leads us to a really crucial regulatory constraint, a disclaimer we absolutely have to make clear for everyone listening based on the source material. These powerful research-grade peptides, like Kisspeptin-10, they are not for general use.
Yes, this is a vital point we have to emphasize strongly. Due to their potency, their effects, their ban status in sport, and, importantly, the lag of extensive clinical trials for safety and efficacy in the general population. These peptides are strictly limited to bona fide research purposes only.
Meaning in controlled labs by qualified scientists.
Precisely. They are not approved medications, they are not supplements, and they are absolutely not intended for human consumption, injection, or any kind of use outside of a rigorously controlled scientific or clinical trial setting. Making that distinction is critical for safety, for legal reasons, and frankly, for ethical research practice.
Okay, let's get even deeper now, right into the molecular machinery. We've talked about the key, Kisspeptin-10, and the lock, Kis1R. But how does that lock, the receptor, actually work at a cellular level? How does it translate that simple binding event into the powerful signal needed to make a GnRH neuron fire?
Okay, so we identified Kis1R as the receptor. It belongs to a very large and incredibly important family of receptors in biology called G-protein-coupled receptors, or GPCRs for short.
GPCRs, right? They're everywhere.
They really are. They act as molecular switches or signal amplifiers on the cell surface. So when Kisspeptin-10 binds to the part of the Kis1R receptor sticking outside the neuron,
The extracellular domain.
correct, that binding causes the receptor to change shape slightly on the inside of the cell membrane, and that shape change activates an associated protein complex called a G protein, which is loosely attached to the receptor on the inside.
Okay, so the G protein is the first domino to fall inside the cell. What happens next? What does the G protein do?
The activated G protein then kicks off a rapid and complex internal signaling cascade using things called secondary messengers. In the case of Kis1R, it often activates an enzyme called phospholipase C.
Phospholipase C. Okay.
This enzyme then quickly generates other important signaling molecules like IP3 and DAG right there at the membrane. And IP3 in particular travels to internal storage compartments within the neuron.
The endoplasmic reticulum.
Usually, yes, and it triggers the release of stored calcium ions, Ca2+, from these compartments into the main body of the neuron, the cytoplasm.
Calcium. That's often the trigger for neurons to fire, isn't it?
Yeah.
The spark plug.
It very often is, yes. This rapid, sharp increase in intracellular calcium concentration, along with other pathways activated by DG, like protein kinase C, PKC, this combination generates a massive excitatory signal within that GnRH neuron.
Enough to push it over the edge.
Enough to strongly depolarize the neuron's membrane potential, pushing it past its firing threshold. And that forces the neuron to fire an action potential, leading directly to that powerful pulse of GnRH being released out of the neuron terminal and into those portal blood vessels.
Wow. And the whole process from Kisspeptin binding to GnRH release. How fast is that?
It's remarkably fast. We're talking seconds to maybe a minute for the full cascade to result in GnRH release. It transmits a signal of extraordinary consequence almost immediately.
So you really see the efficiency. This tiny little 10 amino acid peptide docks onto a GPCR. And within moments, that subtle molecular event unleashes this massive hormonal chain reaction controlling huge physiological systems.
The amplification and efficiency are truly astounding. A tiny pulse of Kisspeptin is all it takes to kick off the whole downstream cascade LH, FSH, sex steroids. But, and this is crucial, that pulse has to be timed perfectly.
Right, the timing, the pulsatility.
Yeah.
That brings us back to the conductor role. Who or what is actually controlling that precise metronomic rhythm, making sure the pulse is just right, especially for sustained fertility?
That critical job falls to a very specialized group of neurons located nearby in the hypothalamus, specifically in the arcuate nucleus. They're often called KNDI neurons.
KNDY. KNDY. What does that stand for?
It's an acronym for three key signaling molecules that these neurons produce and release. Kisspeptin itself, neurokinin B, or NKB, and dynorphin, often abbreviated DYN.
So they make kisspeptin, but also these other two, neurokinin B and dynorphin.
Correct. They co-express all three. And it's the interplay between these three molecules within the KNDNR network that seems to be the critical pulse generator, the timing mechanism.
Okay, this sounds complicated but important. If kisspeptin is the main output signal to the GnRH neurons, what are neurokinin B and dynorphin doing within the KNDA system? How do they create the pulse?
This is where the elegance of the system comes in. They seem to work together as an integrated feedback unit, constantly regulating their own firing pattern to create that pulse.
Yeah.
Think of it like this. Kisspeptin is the primary output signal that goes out to stimulate the GnRH neurons, as we discussed. Neurokinin B, NKB, seems to act as the accelerator within the KNDI network. NKB is a powerful excitatory neurotransmitter. When released, it acts back on the KNDI neurons themselves, making them fire more rapidly. It basically drives the pulse on and makes it strong. Dynorphin, DYN, acts as the brake. Dynorphin is an inhibitory peptide, actually an endogenous opioid. When released, it also acts back on the KND neurons, but it dampens their activity, slowing them down. It effectively shuts the pulse off.
Ah, okay, that makes sense. So within the KNDD system, NKB pushes the on button, driving the kisspeptin release, and then dynorphin hits the off button, stopping it. Creates a cycle.
Exactly. It's this intrinsic push-pull dynamic. NKB stimulates activity, leading to kisspeptin and dynorphin release. Then, the released dynorphin provides negative feedback, inhibiting activity and stopping the release. Then, as dynorphin levels fall, the inhibition wears off, NKB takes over again, and the cycle repeats.
It's like a tiny biological oscillator, a metronome built from opposing chemical signals.
That's a very good way to describe it. And this precise rhythmic on-off cycle generated by the NKB-dynorphin interplay within the KND neurons is what generates the necessary high-frequency patterned release of kisspeptin onto the GnRH neurons.
And if that NKB-dynorphin balance gets messed up,
then the rhythm is broken. If NKB signaling is deficient, the pulse might be too weak or too infrequent. If dynorphin signaling is faulty, the pulse might not turn off properly. Either way, the GnRH pulse pattern becomes erratic or incorrect, and fertility is almost immediately compromised. This KND system is absolutely central.
Okay, and how do these KNDIT timing experts connect back to the bigger picture? Do they get feedback from the rest of the body?
Oh, absolutely. They're constantly being monitored by the downstream products they help create, namely the sex steroids, estrogen, and testosterone.
It's a classic negative feedback loop.
Exactly. When levels of estrogen in women or testosterone in men rise in the blood, these hormones circulate back to the brain and act on receptors located on the KNDI neurons.
And what do they tell the KNDI neurons?
They generally provide negative feedback. High steroid levels tend to dial down KNDI neuron activity, slowing the release of kisspeptin and NKB dynorphin. This prevents the system from overshooting and keeps hormone levels within a healthy range.
And if steroid levels drop too low,
then the negative feedback eases up. The KND9 neurons become less inhibited, they fire more readily, kisspeptin release increases, GnRH pulses increase, and steroid production ramps back up. It's a constant balancing act.
So this complex interplay ensures the rhythm is not only precise minute to minute, thanks to NKB dynorphin, but also appropriate for the body's overall hormonal status and needs over longer timescales, thanks to steroid feedback.
Precisely. It's multiple layers of exquisitely timed regulation. Hashtag outro.
Wow. This has been a genuine deep dive into one incredibly potent molecule. So Okay, let's try and pull it all together. What does this all mean?
Well, I think it means Kisspeptin-10 is just so much more than a simple reproductive hormone. It really stands out as a true master orchestrator of fundamental human biology. It performs these absolutely essential gatekeeping roles.
Yeah.
It controls the literal onset of puberty. It regulates the intricate rhythm of adult fertility using that fascinating KNDI metronome. It clearly links our mood, our desire, our emotional state to our physical readiness.
Mm-hmm.
And maybe most critically for day-to-day life, it acts as that primary sentinel, constantly monitoring our metabolic health and energy status, deciding if conditions are even right to allow reproduction to proceed. Its influence just ripples through everything from our behavior to our long-term hormonal health.
It really is foundational.
Yeah.
Which brings us to our final thought provocation for you, our listeners. We've spent a lot of time talking about how incredibly sensitive kisspeptin signaling is to things like energy balance, stress, overall metabolism.
Very sensitive.
Now, think about modern life. As many people age, metabolic health can decline. Chronic stress is, well, pretty common. And we also know from research that kisspeptin activity tends to naturally decrease somewhat with age anyway. So, the question to ponder is, given everything we're learning about this molecule, what does this tell us about the future potential for interventions? Maybe not just for treating infertility, but potentially for supporting a healthier hormonal environment, perhaps influencing mood, maybe even mitigating some of the metabolic challenges of aging by carefully, thoughtfully modulating the kisspeptin pathway.
That's a really profound question to consider, especially as the search continues for more physiological, more natural ways to support health and well-being across the entire lifespan. Definitely food for thought.
Indeed. Well, thank you for joining us for this deep dive into the world of Kisspeptin-10. And just one final crucial reminder. The compounds and products we've discussed today are potent research substances.
Absolutely.
They are strictly for educational discussion and laboratory research purposes only. They are not intended for human use or consumption in any form. Please stay safe, stay curious, and we'll see you next time on The Deep Dive.